Today we have Hasan DeMircii, who's one of the newer members of BioXFEL. He's a research associate at Stanford University. He's also a member of the Biosciences Division of SLAC and is affiliated with the Pulse Institute at Stanford. And today he'll be giving a talk on SFX with the ribosome.
Thank you so much for the opportunity, and I'm really delighted to give this talk and share some of our recent findings and also some of our old results. So it's going to be more like, giving a summary of what we were doing for the past couple of years. So what really excites me is the ribosomes and the deciphering of the genetic code from DNA to messenger RNA to protein. And we still don't understand how this genetic code is translated into a protein language. Ribosomes are fascinating. They're huge megadalton structures. We call them supramolecular complex. And studying them at molecular detail by using X-ray excites me. When I see ribosomes in action, it's an undescribable joy. So in order to study ribosomes at the physiological temperatures, to bring the biology alive, we have to use SFX. But before that, we have the history of using traditional synchrotron X-ray sources and study these cool phenomena at cryogenic temperatures, which is not really ideal, but I'll give you an overview of the translation, the biology itself, what drives me, what motivates me, and some of our cool results. So in order to do most of these work, I have to collaborate with really phenomenal group of people. At Stanford, we have Tony, Alan, Soichi, and Jody and their phenomenal groups. And recently, we also started collaborating with Michael Levitt and his group. To do some of our LCLS work, the SFX studies, we got tremendous help from Sebastian and his group at LCLS. And also, we carry out some of our cryogenic temperature work at SSRL with Mike Soltis, Aina Kohan, and with the help of Zanko. And also, recently, we studied some of our samples at Sakura. I'll talk about at the end. And at Pulse, when I started, there was Mike Hogan, but now he's left. We still continue to work with Ray. And we have also collaborated at Kumamoto. Cornelius helps us with a lot of SFX work. And originally, I started doing ribosome work and started structured biology of the ribosomes at MRC in Benki's lab. So these are current group from last year. You can see that we work with very young people in a very young field, and you can see the smile on everybody's face because we're doing really exciting stuff. Except Mark, I don't know. This is the best we could have get out of Mark. Especially after beam times, we really achieved impossible things with this incredibly young and ambitious group of people. And yes, indeed, this guy is me with some long hair, with some extra hair and less beard. And Ray is our, not just like the driving force, but also he's the cheerleader of our group. Sometimes, things don't work, but we need Ray to keep things going and also get amazing results. So today I'll talk about the mutant ribosomes and the kind of like the mode of action of antibiotics by using both synchrotron works and also FEL. So briefly, I'll talk about why I study translation, why I want to understand the mode of antibiotics is that we have a very serious problem of infectious diseases, and I would say a very important problem that we're facing today is the rise of antibiotic-resistant bacteria, superbugs. And I'll talk about translation, the protein synthesis, basically the translation of the genetic code to protein language, and briefly talk about the X-ray crystallography technique that I use to study the ribosomes. And I'll tell you about two phenomenal stories. One of them is paromycin, the other one is streptomycin. These are the two cool drugs that helped us so much to learn about the mode of decoding process. And in the end, I'll talk about future, but I work at Stanford. It's like a national laboratory. Here, when we talk about future, that's basically what we're doing today, which is future elsewhere. So you'll see more and more these kind of editorial pieces coming in really reputable journals and newspapers. And this was published a year ago. And here they kind of like explain that if we lose the power of antibiotics, if we don't have an arsenal of antibiotics to fight against the bacteria, against infectious diseases, this will threaten the achievements of all modern medicine that we're using today. So in order to do an open heart surgery or any other basic procedure, basically the patients should have antibiotics. Otherwise, the infection is going to be really a big problem. And we have to have new medicines. And instead of talking all of these infectious diseases, I'll just give it one example to highlight how important and how serious is the problem. For example, tuberculosis. These are the numbers from 2013. There are nearly 10 million people just got infected with TB. And among these people, 1 million were also HIV positive. And again, 1.5 million people died from TB. And people are scared of Ebola, which claimed only 3,000 lives, and it was a news, pretty much every news outlet. Now we have people, millions of people, who are dying from TB, and we don't hear about it. And that's really disheartening. And among those people, 360,000 of them are HIV positive. Why am I just highlighting this? Because people are not dying from HIV. During their suppressed immune system state, if they have any infection, they're destined to die. And when we look at the recent drugs, the frontline drugs that are used to treat TB, the latest one was kind of put in line in like 40 years ago. And it took another 40 years to develop a new and effective antibiotic, betaquilin. So why it takes so long? Because most of these antibiotics work at physiological temperatures, and we can only study these structures. If we want to design a drug based on structure, we have to study them at the physiological temperatures or near physiological temperatures, but we weren't able to do this. So we have to reinvent ourselves to study the ribosomes at near physiological temperatures. And the other disheartening part of antibiotic field is that these drugs are not profitable. And unfortunately, most of the drug companies, big pharma companies, have stockholders, and they don't really invest into antibiotics because they cannot make profit out of it. So it leaves only us, the pharma, basically academia, and some of the small companies to go after these important drugs. So how are we going to deal with the infectious disease? How are we going to target these bugs? We're fortunate enough that our translational machinery is different than these bugs. So here's the central dogma of life. Basically, DNA makes, messenger RNA makes, protein makes us. So ribosome is the most fascinating and complicated machinery that translates the messenger RNA code, the nucleotide code, into the protein language. And compared to both replication and transcription machinery, ribosome makes more mistakes. It's like me. I think in Turkish, and I speak in English, and that forces me to make more mistakes. Either I'm going to talk very slow and make less mistakes, or I'm going to speak at this pace, and you're going to hear a lot of mistakes that I'm making during my presentation. So it's a choice up to you. Either I'm going to talk here for maybe 10 hours and make no mistakes, or you're going to finish in this presentation in 40 minutes, and you're going to hear a lot of mistakes. So here is the DNA language and the structure of DNA. Basically, it's a double-stranded helix. It's very boring structure. And A base pairs with T, and G base pairs with C. It's very simple. If you look at my fingers right now, A and G is slightly bigger than C and T. So what happens is that every time there is A and T and G and C, it's very easy to maintain this double-stranded structure because the chemistry and the stereochemistry and the geometry is very simple. How about RNA? So from DNA to RNA, it's transcription. Again, it's a very efficient or more efficient than replication in terms of error mistakes. And you can see that unlike DNA, RNA has personality. And this is a real structure of an RNA from a ribosome that shows a central pseudoknot. And from RNA, we go to protein language by using ribosomes decoding. So what is ribosome? What does it look like? Here is the magnificent structure of a 2.5 megadalton bacterial ribosome. And you can see that it's a wet noodle soup. It's a big giant mess. It's very complicated, and probably we'll never understand how this machine works. So here on the left, you see the large ribosomal subunit, and on the right, you see the small ribosomal subunit. The small subunit is the brain of the ribosome that deciphers the genetic code, and the large subunit on the left is the hands of the ribosome that links the amino acids together after translation to synthesize the proteins. So what I'm going to do is basically just split these two subunits, and we're going to look at them from the subunit interface to better understand how this machine works. So again, on the left, we see the large ribosomal subunit. This is called the crown view of the 50S large subunit. And on the right, you see the small subunit, and you can see that this view is called the chick view of the ribosomal subunit. And you can see the head, the beak, the body, and the spur of the small ribosomal subunit. So what you see here in these boxes are the sites for the tRNAs. So a tRNA that carries the amino acid, the amino acid that is going to be attached to the protein, enters the ribosome from the acceptor site, and the peptide bonds are formed at the P site, and they move from A to P and exit the ribosome from exit, E sites. So this structure is kind of summarizes the kind of like the direction of the motion of those substrates. So now you look at the structures and probably figure out how this machine works. But just to help you a little bit, I'm going to show you a short movie, and this movie will kind of like put everything in perspective. And this movie is made by Said Sanuga from MRC Laboratory. And this movie combines decades of work that including X-ray crystal structures, cryo-EM structures. And here's the small ribosomal subunit. Messenger RNA is also shown here. And the first code is deciphered by a very special tRNA, formulated methionine tRNA, FMET tRNA. And it also gets help from 3 initiation factors to make sure that the code is in frame with the tRNA. After the subunit joining, the initiation factors dissociate from the ribosome. And now ribosome is ready for elongation phase. So it switches from initiation to elongation after the first alignment of the tRNA with the messenger RNA. So tRNA is delivered to a ribosome by a factor called EFTU. After GTP hydrolysis, the tRNAs are delivered, and the peptide bonds are formed on the large subunit at the peptidyl transfer center. After the peptidyl bond is formed, a second factor, EFG, comes, hydrolyzes energy, and translocate the messenger RNA and the tRNA on the small subunit to open up the acceptor site for the next tRNA. And this process happens 20 times per second. So every 50 milliseconds, ribosome accepts another tRNA and decodes it and attaches that one amino acid to the nascent polypeptide at the peptidyl transfer center. And then the nascent polypeptide exits the ribosome from the exit tunnel. And here you can see a very low budget of protein folding movie. And now after the proteins are synthesized, the elongation phase comes to an end. And once ribosome encounters a stop codon at the A site, a molecular Caesar called RF, either 1 or 2, comes and clips that nascent polypeptide, and ribosome finishes the translation and ready to be recycled. And this is accomplished by a recycling factor in another molecule of EFG. So this primes the ribosome for the next round of elongation. You can see that ribosome is very fast and quite accurate. And then the question is that how we're going to study these molecules and understand the mutations, antibiotic resistance, and decoding, and all of these processes at the molecular level. The technique we call this X-ray crystallography. basically what we do is we accelerate or decelerate electrons to generate X-rays, and then we focus our X-rays onto a crystal and collect the diffraction image onto a two-dimensional detector. So what literally happens is that a three-dimensional object is flattened into 2D information. So we grow crystals, and the crystals come in different shapes, different geometries, not in different colors. These are the kind of the tricks that are created by the polarization. They're beautiful. They're incredibly beautiful. And out of these beautiful crystals, we get beautiful structures. So here, basically, this slide summarizes how human eye can see things. So if there's a large plaster object, a model of a ribosome, an incident light reflects from the surface of this object, and the human eye can focus this visible light, or UV visible range, into the human retina. And then we can see the image flip out, and then we can create those images in the brain, the optical center. So how do we see small objects? In order to see small objects, we have to use very small wavelength, not the UV visible range. And the best part of the light spectrum is the X-rays. And we can use hard X-rays in the 1 and 2 angstrom range. And this gives us enough resolution to resolve individual atoms. And what happens is that if you have a ribosome crystal like this, the incident X-rays diffract from these molecules into a, I would say, two-dimensional pixel detector. Unlike visible range, there is no lens that can focus the X-rays. So that's why we have to do some magic. So basically what we do is we take these two-dimensional diffraction image and use some prior knowledge, the sequence, and some other knowledge about the object that we study, and use some algorithms to create these three-dimensional objects. So this algorithm box here is representing the X-ray crystallography. So there's so many formulas that has to be crunched, and then we create these experimental electron densities. What we basically do, as I mentioned before, we have a prior knowledge. What it means, we know the sequence of these objects, but we first trace the main chain of the proteins or nucleotides and look at the additional protruding electron densities where we start building the remainder of the molecule. And as a result, we have an atomic model where we can study the objects in unprecedented detail. So what I'm really interested in is how the antibiotics work and how they help us to understand the decoding process. And there are also mutations on ribosome that use either defects in decoding or antibiotic resistance or dependence. So here's the genetic code. This is bacterial genetic code. You can see that there are certain peculiar things about these codes. For example, if you look at just these degenerate codon box here, you can see that ribosome can easily discriminate between first and second letter. It's always very accurate, and it doesn't really matter on the third position what nucleotide is positioned. It's always codes for this amino acid called serine. So how does ribosome discriminates between first, second, and third position? So decoding, the deciphering the code is quite fast. It's 20 decoding per second, and it's quite accurate. Ribosome makes 1 in 1,000 to 10,000 peptides linkage, and this accuracy is accomplished by a two-step kinetic proof reading model. So what happens during this two-step proof kinetic reading model, ribosome goes through a series of steps. I know this slide looks very simple, but I promise that I'm going to make it even more simpler, taking you through each of these individual steps structurally. So what happens in the first stage, ribosome receives the first tRNA to the A site. And to simplify everything, I excluded all the ribosomes and just showing you the messenger RNA. And the purple here is, or magenta is the first code in the A site. And this is the position 1, 2, and 3. And messenger RNA goes from 5' to 3'. That's the directionality of the reading. And the tRNA aligns with the message in the A site. And ribosome has to monitor this codon and anticodon base pairing. How does it monitor? As I mentioned before, A and T and G and C base pairs and creates this isosteric same distance. The iso means same, steric means geometry. So if A and G, U and C base pairs, then you see the distortion. And this thing is kind of like the basis of the decoding. So ribosome basically measures this same geometry every and each time you have a codon, anticodon base pairs in the A site. So ribosomal decoding center consists of RNA, exclusively RNA, and the key decoding residues are here, and they flip out to monitor the first and second position, and these are like the fingers that just like fits into a glove beautifully if there is correct codon-anticodon base pairing in the A site right here. So the third position is monitored by this residue called G530 by an angle. So when you have this angle, you lose the precision that you achieve in the first and second position. So after this monitoring, ribosome goes into the next stage. So the question is that, how are we gonna accommodate this correct tRNA? So this correct base pairing induces some additional conformational changes that brings us to this GTP activation. So now I'm gonna put everything into the context of the ribosome. I'm gonna zoom out, and now we're looking at the codon-anticodon in the A site of the ribosome. So the green part is the brain of the ribosome that decodes that codon-anticodon base pairing. And what we're gonna do is we're gonna chop the head of the ribosome through this plane that I've shown here, and we're gonna look at it from the top angle. So now you can see that ribosome goes from an open to closed transition. So when there is no tRNA in the A site, it's open. And when there is tRNA codon-anticodon base pairing, those two critical brain residues flip out, monitors the same isosteric geometry, and once that is monitored and kind of like proven that it's the right correct geometry, then ribosome closes on top of that correct base pair. And then there are mutations that makes the ribosome either more error prone. Instead of making one mistake in 1,000 to 10,000, ribosome starts making more mistakes if we have mutations in this error prone region of the ribosome that favors the closed state. And there are also mutations on the other side, we call them restrictive mutations, that favors the more open state of the ribosome, and now ribosome becomes more accurate. If you remember my analogy, I said I'm also translating. It's all about speed versus accuracy. So ribosome is optimized for this open-closed transition to achieve that accuracy at certain speed. So if you want to make ribosome more accurate, like restrictive phenotypes, they make less mistakes, but also they become very slow. So I'll talk about those mutations later on. So now if you put the ribosome back into the original state where we started and flipped out, you can see that that open-closed transition comes from screen towards you. And if you put the A site tRNA, P site tRNA, and the E site tRNA on the ribosome, and if you remember that the A site tRNAs are delivered by an elongation factor, that domain closure induces that release of this factor from the correct tRNA, if it's correct tRNA. If it's not correct tRNA, then there is no domain closure, and ribosome rejects that tRNA and looks for the next tRNA. So then that brings us to GTPase activation and accommodation. So that domain closure induces GTPase activation on the factor. And if you look at the entire complex, all ribosome just as a single piece, after the GTPase activation, the A site tRNA has two options. Either it's going to go and accommodate it in the peptidal transfer center and catalyze the peptidal transfer, or it'll be rejected during the proofreading stage, and the next tRNA will come. So then the question again is, how antibiotics and mutations will help us to understand this process of decoding. So this is a quote from a very important book. This is the Bible of antibiotics field. And in this book, which is written 40 years ago, I think it's still a phenomenal piece of art. These famous people collectively tell in the antibiotics section that targets the ribosome, the diversity of this effects produced by this aminoglycoside, and the ingenuity with which they have been pursued have made the streptomycin story the most fascinating and complicated one in the field of anti-ribosomal action. So I promised you that I'm going to talk about the streptomycin story, and these people are telling you that it's going to be a fascinating story. And this is also a kind of a landmark paper published, again, decades ago, and show that ribosome has a decoding site that senses codon-anticodon base pairing, and the drug streptomycin induces errors in this process. So when I started my research, there was only one ribosome structure which is bound to streptomycin, but that same structure had other two drugs, namely parmomycin and spectinomycin. So it was complicating everything, and we have slurry of genetics and kinetics data and biochemistry data was kind of conflicting with those results. And basically, in this paper, what these people showed is that the ribosome can effectively discriminate between correct cognate and incorrect tRNA on the ribosome. If we give parmomycin, another aminoglycoside that binds near this decoding center, without altering the selection of the correct tRNA, parmomycin increases the decoding of the non-cognate or incorrect tRNAs in the ribosome. And this kind of causes a discrimination errors in the bacteria, and instead of incorporating the correct tRNA, they start inducing or incorporating wrong amino acids to their proteins and fill the entire cell with nonsense proteins. And when we look at the streptomycin, unlike parmomycin, reduces the discrimination of the correct tRNA and increases the incorporation of the incorrect tRNA and puts the ribosome into a complete indiscrimination. So this drug is literally causes suicides of the bacteria. Then how are we going to discriminate the impact of the streptomycin and separate from the action of parmomycin if you don't have a structure that has only streptomycin? So that's why we solve the structure. And when we look at the details, the gray structure is the apo, empty ribosome, and the green one is the drug-bound structure. You can see that when streptomycin binds, it distorts the brain part of the ribosome where the residues 1492 and 1493 is located. So those two residues has to flip with a perfect precision to look at the codon-anticodon base pairing. So if you distort that geometry when those two residues flip, they're not going to be monitoring the codon-anticodon base pairing, but literally just putting ribosome in a very indiscriminate state. So streptomycin literally destroys the brain of the ribosome. So when we look at the electron density of the ribosome program with the cognate tRNA and the messenger RNA, we see a nice and well-defined electron density. When the same cognate tRNA is given to ribosome programmed with the streptomycin, we see less binding. And that was kind of consistent with the biochemistry data. And we also find out that in this state of decoding, ribosome not really induce the complete domain closure. And that's why ribosome loses their efficiency of decoding the correct tRNA like an incorrect tRNA. So if I summarize everything that we know about streptomycin structurally, I'll show this movie. The drug binds right at the heart of the ribosome near the decoding center. And when the drug binds, it induces a lot of conformational changes locally and globally. So the global conformational changes are irrelevant because there are a lot of crystal contacts. But if you look at the decoding center, the binding of the drug distorts the decoding center, especially these two critical residues here that deciphers the first and second position of the code. And in addition, the decoding center is stabilized by this hydrogen bonding network from the neighboring helix of the ribosome called helix 45. And these network of hydrogen bonds are broken when the drug is bound right here. So that makes a very floppy decoding center, a mushy, it's like a marshmallow, softened up decoding center, and kind of makes the ribosome to lose their discrimination of the correct and incorrect tRNAs. So the streptomycin basically, if I summarize, distorts the decoding center. And now I want to talk about a very interesting phenomenon and talk about this, not resistance, but the dependent phenotypes of the mutation. So there are ribosomes, they are literally dependent on the drug streptomycin. So you give the drug to a wild type bacteria and they're sensitive and they die. And some of the bacteria picks up a mutation and becomes resistant to this drug streptomycin. And when you take the drug off, they die. So without knowing, they become dependent on the drugs. So they become junky ribosomes. So these are the wild type crystal forms of the wild type ribosomes. And when we crystallize the mutant, the streptomycin dependent ribosome, we obtain these crystals. First, we thought that they're salt crystals and maybe like they didn't crystallize. And when we take them to the synchrotrons, they diffracted like ribosomes, so we even saw the structure. But what's so fascinating about these two crystal forms is that by just looking at the crystal morphology, you can see which one is active and functional, and which crystal form is inactive and dependent on the drug streptomycin. So when we crystallize the ribosome, these dependent ribosome with the drug streptomycin, we obtain, again, the wild-type crystal form. And sometimes bacteria picks up a second suppressor mutation that suppresses the dependent phenotype, and they also crystallize like wild-type. So when we look at the crystal packing of the wild-type versus dependent mutant ribosomes, you can see a slight, I would say, a radical shift of this crystal packing. The beak of the ribosome, instead of hanging here, it interacts with this blue protein that is sitting here. And this protein now interacts with the other beak. So there's a remarkable shift in the crystal packing. So just to summarize what happens in the decoding center and why these ribosomes are dependent on this drug, I want to emphasize how small is the mutation. We're looking at megadalton structure, and the only change is this proline residue is mutated to a tryptophan. So it's a single amino acid chain in the entire ribosome that favors this new crystal form. And when we calculated the experimental electron density, we're looking here unbiased omit map, and you can see an extra piece of density that belongs to that mutation. And when we model the new structure, you can see that that tryptophan also caused radical conformational changes in the decoding center of the ribosome. So then the question is that, why they become dependent on this drug? So if you remember, the streptomycin was pulling this decoding center towards left. And this mutation is pushing the decoding center residues 1493 and 1492 in the opposite direction. And there's a tug of war. So the drug wants to pull it to the left, and the mutation pushes to the right. And if you don't have drug, you have a very distorted brain or damaged brain, and these ribosomes are non-functional and it's lethal. But if you give them the drug, it pushes back to the left side, but then there's a mutation pushing it in the opposite direction, and the structure is somewhere in the middle. That's where the wild-type ribosomes work. So hopefully these images explains why ribosomes become dependent on the streptomycin, and also help us to better understand the decoding mechanism. So if I summarize the dependent ribosomes, the dependent mutation causes extreme distortion of the decoding center, and when the drug is present, it partially restores that deformation of the decoding center and provides kind of like a partially active ribosome and kind of helps the bacteria to survive in the presence of the drug. So now I'm going to switch gear and talk about why we switch from cryogenic temperature studies to near physiological temperature studies. So I work at SLAC, and LCLS, the linear coherent light source, is the world's first hard X-ray free electron laser located here. And our lab is at the Pulse Institute, right behind here. And we do some of our research at the CXI end station of the LCLS. So when I first started doing ribosome SFX studies, the method was described to me as diffraction before destruction. So literally you put, these are single molecules, but imagine introducing crystals. The short pulses of the LCLS can get in and get out of the sample, and the data will be collected before it's damaged. So technically, we can outrun the radiation damage. And that was quite exciting. And at the time, I was working at Brown as a postdoc, and I decided to bring some of my ribosome. And this was kind of like 4 years of literally nonstop purifying, crystallizing, harvesting ribosome at 13 milliliter, a record amount of ribosome crystallized elsewhere. So then we thought that 13 milliliter of ribosome slurry that I brought from Brown to Stanford was going to provide us with the first room temperature crystal structure of the ribosome. And this is basically what has been always presented in the previous publications. An injector, a light source, and a detector. But I didn't know at the time that we have to also do some preliminary filtration of our crystals and do it multiple times, and including one inline filtration during the sample injection. So ribosomes are very delicate. Unlike lysozyme crystals, even if you sneeze near these crystals, they lose their diffraction quality. So imagine now these filters as what I would describe as a meat grinder. Put a steak from on top, and what you're going to get a ground beef. Yes, the technique is called diffraction before destruction, but these filtrations, due to those tiny capillaries that filters has to filter out those big chunks, destroys the sample. So diffraction before destruction becomes destruction before injection. So in order to study ribosomes, we have to come up with a better method, and that forced us to reinvent some of the existing methods. And thanks to Mike Bogan and an amazing grad student at the time, now he's a research associate at LCS, Ray Sierra, they had this electrospinning method to introduce the crystals of the biological samples to the LCLS or any light source. And what we did is we kind of improved this method. Here you see a single capillary, and this method only worked at the time because it was in vacuum. You have to dope these samples with the cryoprotectants, and technically we can't really alter the crystal slurry where the mother liquor is, because if you do that, ribosome crystals are going to lose again their diffraction quality. So we wanted to develop a method where we didn't have to change where the crystals of the ribosome. We don't have to filter them because we can use a very large capillary. And in the previous method, we used 13 milliliter of sample and get a beauty. That's what they called it, a beauty shot, a 6 angstrom diffraction pattern. And we basically get maybe like 10 of them, and that was a dead end. But now we use, instead of 13 milliliter of sample, we use less than half a milliliter, technically like 300 microliter of sample during 6 hours of screening time. And the only difference that we employed for the electrospinning method is to this $10 piece of McMaster T-junction. And now the ribosomes are directly goes all the way to the interaction region of the X-rays, and they're uninterrupted, unfiltered. And we also provided the cryoprotection from a second line, which is also kind of charged with electricity. And now we can protect our crystals all the way to the interaction region without changing the mother liquor. At the time, the previous users also destroyed a quarter of the detector. So now we use less sample, less detector, and less time, and we obtain a better first room temperature structure of the ribosome. And here's the method. This is the electrospin injector. It goes all the way down to the interaction region by the pulses of the LCLS and produces these amazing room temperature structures of any kind. So here what I'm showing is the first ambient temperature structure of the ribosomal subunit, 30S, bound to drug paromomycin. So paromomycin is a very cool drug because we learned so much about decoding because of the parmomycin. And you can see in blue color the cryogenic temperature of the 30th structure that we determined at synchrotron. And the light color brown structure is the first SFX structure that we obtained at LCLS to similar resolution and similar R factors, R and R3. If you look at these structures, basically for an untrained eye, they look very similar. But when we focus on the drug binding pocket and also another region of the ribosome that we describe as helix 28 or hinge region, you see a quite remarkable shifts. So when we focus on the drug binding side, one of our goals here at Stanford is to develop better aminoglycosides that target ribosomes. And parmomycin structure here showed us that there are significant changes when we look at the drug binding at room temperature versus cryogenic temperature. And also, this provides like the basis of a better starting model to develop better drugs. And I'm going to show more examples that we recently obtained at LCLS. And when we align the entire ribosomes and look at the RMSDs of the backbone residues, you can see that it's a very similar alignment throughout, except the big part, which is a crystal contact, and that hinge region that I mentioned, the helix 28. So the helix 28 has been kind of like the part of the ribosome that provides the remarkable plasticity to the ribosome during decoding. I showed the movie. Ribosome has to go from state to state during decoding. So it has to go kind of translocation. It has to accommodate the tRNAs, the substrates, the factors, initiation factors. And this hinge motion literally gives the plasticity or the flexibility to the head and allows ribosome to shift from state to state. And Harry Noller, a towering figure in the ribosome field, superposed all the existing structures of the ribosome and published a paper in PNAS and literally saying that helix 28 provides that plasticity to the ribosome. But here all we did was to study the structure of ribosome at room temperature and cryogenic temperature without changing anything else. So literally by looking at the temperature fluctuations, we were able to see this kind of hinge conformational change, and that kind of like popped up very naturally. So the RNA bases are stacked on top of each other, and these base stackings are very dependent on the temperature shifts. And this kind of provides, again, another reason why some of the structures can be more accurate when they're studied at room temperature. So these results are kind of like really promising and kind of like motivated us to do more on these fields. And Tony Ricci reached out to me and he said, like, we have these new antibiotics. They are more potent and antibacterial, and they have less ototoxicity. I said antibiotic field kind of like now is completely abandoned by the pharmaceutical companies. And we also joined forces with the Michael Levitt and his group, especially Fred, to do some of the computational analysis to develop and design a new generation antibiotics by using room temperature and cryogenic temperature crystallography of the ribosomes. So the goal is to develop a drug that has no side effects. Why side effects are so important? For example, when a kind of prematurely born baby is placed into a neonatal intensive care unit, they're very prone to getting infected. And when the doctors see these infections, they have to use a very broad spectrum antibiotics. The most broad spectrum antibiotics are aminoglycosides because they target the decoding center of the bacteria, and it's universally conserved center. But the problem is that the same drug also targets the hearing cells and causes hearing defects. And these babies are given aminoglycosides, and then when they discharge from those neonatal NICU centers, they're not tested for their hearing. And then you can see a lot of kids walking around with hearing device. Probably they are the ones that are treated with the aminoglycoside during that hospitalization period. So now we have the opportunity to use the FELs, LCLS, and other FELs to study these things at room temperature and come up with better drugs. So what I'm showing here is some of our unpublished results. And this is a drug called, maybe I shouldn't even give the name. This is a parent compound that we studied. And then we studied at kind of cryogenic temperature. And this shows like we were able to obtain the electron density for this compound. And there was another crystal structure in the PDB, but that was obtained by a small fragment of the RNA, not really the whole ribosome. And when we look at this structure, you can see that, again, significant differences in the decoding center. It's not really an accurate model to start with modifying this drug. And then what we did next, we also studied the same compound by using FEL, and we superposed those two structures, and literally at room temperature, which is the blue structure, and the cryogenic temperature structure, which is the gray, they're quite similar. So this gives us further confidence that we can screen some of these compounds at synchrotrons. But when we study the, or when we look at the neighboring part of the decoding center, this compound binds at the decoding center right here, but has also this conformational flexibility in the neighboring region. So when the ribosomes are in decoding state, they have to be completely in this decoding conformation. But this drug causes misreading without kind of putting the ribosome in the fully decoding state. So it's a very novel mode of action for a compound that causes misreading. And that flexibility, that kind of plasticity or the dynamics was maintained even at room temperature. So we didn't really see any difference when we study the entire decoding center. So we get a very accurate picture of the decoding state of the ribosome when we study it at cryogenic temperature or at room temperature. But the new compound, which is a derivative of the same compound, when we studied at cryogenic temperature, excuse me. So when we study this new derivative compound at cryogenic temperature and superpose and compare with the parent compound, again, we didn't see much difference. So then the question was that how this compound can be less autotoxic or a better compound. So then we studied at cryogenic temperature to room temperature. Again, we obtained a beautiful electron density map for the compound, including the density for the modification. And we superposed the cryogenic temperature over the room temperature structure. Again, we didn't see any difference. So this was puzzling, but the answer came when we look at, again, the decoding center near the helix 45, near the decoding center. So this part of the ribosomes, again, has this very conserved sequence. And there are two modifications here, methylation of the two adjacent adenines, that kind of puts the ribosome from these dynamic states. When we study the parent compound and modified compound at cryogenic temperature, we have this flexibility, but this modified compound at cryogenic temperature, despite the fact that didn't alter this conformational heterogeneity, at room temperature, it provided us with this single decoding state. So that kind of provided the hint why they are less autotoxic. So this part of the ribosome also implicated in the autotoxicity or the hearing loss in the humans. So by looking at these compounds and their structures at cryogenic temperature, we were not able to get this information, but at room temperature, we get this extra piece. So once we get this information, now we have the opportunity to study the next compounds, the next important lead compounds at room temperature, so that we have the further confidence that even before go further clinical trials, they can be just understand easily by looking at this dynamic information here, whether they're less autotoxic and worth pursuing, or maybe it's still very autotoxic and they should be discarded, and we should further develop next compounds with new modifications. So these are our latest results on the antibiotic discovery front, and also our injector technology provided us with new opportunities. And during this screening time, we were able to not just solve one ribosome structure, but we also collected diffraction data from the large ribosomal subunit in record time by using record low amount of sample in liquid state without even altering the mother liquor of the ribosome. So the 50S ribosomal subunit were never able to determine at cryogenic temperature by using our strain, the Thermus thermophilus. And the reason for that is that if you take these crystals to synchrotron, after the first shot, there is no diffraction. And the first shot, even after a few seconds, goes to like 30 angstrom, and maximum you can get is like 15 to 10 angstrom range. And by using LCLS, we have shown that these crystals has potential to diffract beyond 4 angstrom. So we collected a diffraction data extending to 3.5 in this very short amount of time by using our injector technology. And this is the first room temperature structure of the large ribosomal subunit that excites us even further, because most of the blockbuster aminoglycos antibiotics such as macrolides, ketolides, tuberactinomycins, they target the large ribosomal subunits. And now we have the luxury to use our large and small ribosomal subunit crystals to study all of these exciting antibiotics and the new generation compounds at physiological temperatures to push the further development of the new antibiotics that are more potent, less ototoxic, and save even more human lives. And I said that this technology is quite fascinating and exciting, and I showed this image maybe a couple of times. But the idea is that during my short tenure here, what I have seen is that the SFX field has many injectors for many different samples. So if you have a sample, they develop an injector. You have another sample, they have to develop another injector. For liquid sample, use this injector. For heavy viscous sample, use this injector. For LCP and lipidic cubic phase, sponge phase, use this injector. The idea was also, while we're doing all of these exciting research, also push the limits of the innovation. And we discovered that this technology uses less sample than any other injector and more versatile and almost universal and capable of delivering any sample to any light source. So what I mean is that basically we created a load of the injector that can basically do any SFX or serial crystallography, even at synchrotron, without any effort. And the best part, it's cheap and can be built by a non-expert. Some of the young people that I showed early in my talk was the builders of those injectors, and they literally come freshmen, some of them are like sophomores, juniors, and they come to our lab for an internship, and they are mastering building these injectors, using it, collecting data, and solving those amazing ribosome structures with cool antibiotics. So recently, we also took this injector to Sakura, the Japanese FEL, and we collected some data from, again, small proteins in liquid, in air. They weren't in vacuum. So then we were able to collect, again, structure after structure in a very short time. It was like eating potato chips. So they were so exciting and come one after the other. So we also show that our injector, which is developed by Ray Sierra, is not only works at liquid phase in vacuum, it's also capable of working in air, in liquid sample, by monoline, LCP. We even see a monoline in the resulting structure. And then that even further gave us confidence that it may work at synchrotron. Again, you see a lot of happy faces here. We were able to solve structures even better resolution by using the FELs. And then also at synchrotron, we matched the resolution of an FEL, and we solved more structure by using synchrotrons. And now that opens up this further for general community, because a lot of people, as far as I know, they're kind of hesitant to do serial crystallography, not just SFX, but even SMX. So now both serial crystallography, serial femtosecond crystallography, and serial millisecond crystallography will be available to non-experts by using this technology. And hopefully this will enable really exciting science. We don't have to worry about the technology too much, but just focus on our science. What really drives us? What really we're passionate about? And not worry about the technical details. And I hope that the future is very bright for everybody, not just for selected, a few privileged groups who are capable of doing this kind of research. And with that, I would like to thank you all, and thank you for your attention. And if you have any questions, I'll be very happy to answer. Thank you.
All right. Thank you, Hasan. That was a very good presentation.
Thank you.
Do you have any questions? Preferably from any of the students. Anyone? Well, I have a few questions if no one else does. So I'm curious as to how you're able to get this much ribosome. You said this is from Thermophilus, so assuming you grow up bacteria and then harvest it somehow, but how do you ensure that you have a homogeneous sample? There's a lot of pieces to this.
Yes, so that's a trade secret. So
oh, okay.
it takes us like almost 2 weeks from start to finish to make like a few hundred microliter of ribosome. It's an amazing recipe which is developed by Venkaramakrishnan, and it's also very well documented, published work, but you have to be very meticulous. And every step of this purification protocol has to be executed to perfection. And I think we kind of like mastered that protocol thanks to Venki. But again, there are only a handful of labs that can do those kind of purifications. In terms of like scalability, I believe that right now, currently, our lab at Pulse Institute is the largest scale ribosome laboratory that can purify the amount of ribosomes needed for the original SFX. But now we reduce the sample consumption so much. Now anybody who can do ribosome purification can do ribosome serial crystallography at either LCLS or SACRA. So it shouldn't be a problem. Or European XFEL.
Yeah. Cool, thanks. So I was also wondering if you'd considered possibility of doing some type of single particle imaging with this, maybe something time resolved. And do you think that it's even possible to do that? I think it'd be really
that's
cool.
a great question. Like I said, it's an emerging field. Single particle imaging is also like we're part of the single particle initiative with Ray, and there are discussions about using this technology for single particle imaging too. And I think, as far as I know, if there is willingness to use it, there is always room for improvement, right? If you keep doing the same thing and expect a different result, that doesn't make any sense. So far, I don't think electrospin injector was literally just like too much used in these single particle studies. But as far as I know, it can literally may yield or provide better results. And there are tests has been done by Ray in-house in our lab, and it's a very robust and very versatile technology. And I think in the future, we're going to see some of the single particle experiments that has been carried by the electrospin technology.
Yeah.
Because another thing is that, we're doing electrospinning, but once it spins and forms that cone, after the Taylor cone, it starts spraying. So a lot of like mass spectroscopy has been also done by electrospraying. So this technology has been like tested intensively for these kind of research, but somehow like had a limited usage in the single particle imaging. But I hope the future is again, kind of, to me, it's bright for single particle initiative and also electrospin technology.
Yeah, great. Thank you. Any other questions for anyone? No? All right, well, thank you again for the great presentation. We should have this up fairly soon. We have to do some processing of the video, but we'll have it online on our website and also on our YouTube channel. And for any of the students that missed it, I'm sure they'll be able to catch it later. So
again, thank you for the opportunity. It was a great pleasure.
Yep.
Thank you.
Nice.